drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. Hey everyone, I want to share with you a new AI-powered research tool I've been using to find scientific papers and learn from the literature. It's called Consensus. It is an AI-powered search tool built using large language models trained to help you find and understand the best science faster. Consensus helps you not only find individual papers, but its AI models will help you assess research quality and even synthesize insights across multiple papers for you. You can ask it general questions about any topic you might have, such as what are the known risks and benefits of a certain prescription drug? What do we know about how a certain food or nutrient affects the human body? Anything you can think of. Its Pro Analysis feature will actually provide you with a concise, written summary of what the literature says, broken down point by point, highlighting the strength of the evidence across papers, and providing you with individual studies ranked by the relevance and including additional information like citation rates, journal quality, and more. You can also save, cite, and share individual studies with a single click. Unlike other AI and search tools like ChatGPT, PubMed, or Google Scholar, Consensus uses over 20 custom large language models that are developed and trained for this specific purpose. The base Consensus product is totally free, but the premium product that you can subscribe to has better features and unlimited queries. And I highly recommend this if you are using outdated tools like PubMed or Google Scholar, or you just do your own research. For a limited time, you can use the code MINDMATTERSPECIAL, all one word, to get a free premium subscription for 1 year. That is a $150 value for free if you use the code MINDMATTERSPECIAL today. So click the link in the episode description or check my support page and give Consensus a try. Use the code MINDMATTERSPECIAL to unlock your free premium subscription today.
So I'm Rose Bagot. I'm an associate professor in the Department of Psychology at McGill University in Montreal, Canada. I'm also an associate member of the Department of Psychiatry and a primary investigator in the Ludmer Center for Neuroinformatics and Mental Health, and a member of the Concordia Center for Studies in Behavioural Neurobiology.
Mm-hmm. And so what does your lab do in terms of, you guys study animals? Do you study humans? Are you doing molecular stuff, system stuff? Where do you kind of fall?
Yeah, so almost everything we do is in mice. And in terms of what we do, I'd say that broadly, we're interested in understanding the neural bases of affect and affective behavior. But as I said, we're doing this in mice. And sort of the approach that we take is starting with robust behavioral paradigms in mice. And then we use this as a foundation to explore the molecular, cellular, and synaptic changes that are induced by things like chronic stress or other kinds of experiences, and to understand how that changes neural circuits to lead to changes in behavior. Mm-hmm. So we've done a lot of work looking at how chronic stress leads to changes in the brain with sort of differential emergence of susceptibility or resilience, and using this as sort of a window into understanding mechanisms of stress-related disorders like depression.
Mm-hmm, mm-hmm. And, starting at a very high level, if we think about just animals generally, a little mouse, a human, whatever, just mammals, birds, like just animals in general, we all experience stressors in life. It's just part of being a living thing. When we think about sort of the natural, healthy, ecologically appropriate mammalian stress response, can you just walk us through sort of the basic stress response as it should work under normal circumstances in a healthy individual? What are some of the hallmarks of that?
Yeah, so I'll answer that directly in a second, but I think the really first thing to say is this idea of like a normal or adaptive stress response. There is no absolute. It's really contextually determined. Mm-hmm. And I think that's just sort of generally an important sort of caveat to keep in mind. But in terms of like what is the stress response, at the simplest level, we can talk about the HPA axis, the hypothalamic-pituitary-adrenal axis. And so when an organism, be it sort of me joining a podcast or a mouse in the lab, encounters some kind of stressor, so something that elicits a stress response, we get activation of the HPA axis. And this leads to release of various hormones that ultimately leads to release of cortisol in humans, corticosterone in rodents.
Okay, so we can think of the quintessential stress response as being a measure of how much this HPA, this hypothalamic pituitary adrenal axis, gets activated. And when we say activated, we basically mean stuff is happening in that part of the brain such that a certain cocktail of hormones get released.
Yeah, in a sense we can. And so this is what we mean by sort of the classical stress response, is this HPA axis activation. But most of what we study in my lab is, you could consider it as being sort of upstream of the HPA axis. So we're interested in what's happening in sort of higher brain regions that influence how the HPA axis is being recruited.
Yeah. Yeah. So like if I'm out in the wild and a tiger jumps out of me, my HPA axis will light up. I will have a cortisol spike and all that. But, I had to see the tiger. It had to go through my retina, get in somewhere into my nervous system before that stress response got elicited. And so there's sort of probably multiple things happening in parallel here, from the activation of the HPA axis itself to the object recognition and the sensory perception and all that.
Yeah. And so the tiger jumping out at you in the wild is a pretty unambiguous stimulus. And so then you really want this sort of like very acute activation. And there are sort of other systems that are responding to that as well. But a lot of what we're interested in studying is probably more in what you consider the gray areas. And so how stimuli that could be threatening or maybe aren't so threatening, how that could lead to changes in behavior. So I'd say that in a sense, like the HPA axis is in the background of a lot of what we do, but it hasn't been the focus of anything I've done since back in my PhD, where we really were looking sort of more directly at HPA axis regulation. We did some work that the lab where I did my PhD, we were looking at how just very small variations in experience during the first week of life in rats, and the experience was how much they were licked by their mother. How that leads to changes in specifically how the HPA axis is regulated in response to a stressor. And we can look at sort of the time to the peak hormonal response. And then also a really critical factor in HPA axis function is the shutdown, because you need to have this strong response to a stressor. You need to be able to mount a response. But then when that threat has dissipated, you need to be able to come back to baseline.
Yeah. And I imagine there's a lot of variability between species and between individuals and that stuff, how quickly it comes on, how quickly it goes off.
Yeah.
Whether or not it comes on in response to something that someone else might perceive as innocuous, you perceive as a huge stress.
Yeah, because yes, you can also have disorders of HPA axis function where you don't mount an appropriate stress response. So there's lots of ways that the HPA axis can go awry.
Yeah. Before getting into some of that and some of the neural stuff, I want to cover off on a couple other basic things. So we mentioned the HPA axis, and one of the key outputs of that is cortisol. Cortisol is pretty widely known. Most or many people out there have heard of cortisol. We call it the stress hormone or a stress hormone. What exactly makes it a stress hormone in the sense of like, what is the cortisol doing? What kind of physiological changes does it instigate?
Yeah, so cortisol in humans, or corticosterone in rodents, binds two different receptors, glucocorticoid receptors and mineralocorticoid receptors. And these receptors are widely expressed throughout the brain, but also throughout the body. And so what does CORT do? Many things. So acutely, it's really important in kind of reorienting the organism towards meeting an acute challenge. So you get changes in metabolism, changes in alertness, but chronically you get a lot of sort of negative consequences where you have this continued activation and continued stress response. So you can get like metabolic disorders. You can even, in extreme cases, have stunting of growth. You have a shutdown of reproductive functions, and you can have atrophy in various brain regions, but also hypertrophy in other regions. So it has very wide-ranging consequences.
Mm-hmm. But among other things, it sounds like the cortisol, it's having hormonal effects that influence how the animal's mobilizing its energy stores.
Yes,
for example.
for sure. Yeah, that's a core function.
Interesting. Yeah. So instead of, so if it's a growing animal, for example, some fairly large percentage of your energy is going to go towards growth and building new tissues and stuff. But if you're stressed a lot, you're going to mobilize that for other purposes, and then therefore you're going to take away from that type of function.
Yeah, absolutely.
And when we think about this on the neural side, I'll let you sort of navigate us in terms of some of the experiments that you've done in your lab that are relevant. But, when an animal is perceiving a stressor, before maybe even the HPA axis turns on, what are some of the major neural pathways that are involved in, if it's threat detection or just the perception of a stressor that's, before that hormonal effects of the HPA axis actually start?
So really. So upstream, we're talking about upstream of the hypothalamus. So the hypothalamus is a brain region that's the sort of, you could think of it as the start of the HPA axis. It's the H in HPA. And the hypothalamus receives inputs from many different brain regions. And in terms of what brain regions are going to be processing the different threats, that would depend a lot on the nature of the stressor. And so you could have sort of direct inputs from the amygdala related to sort of like fairly unambiguous threats. We've done a lot of work looking at, I'd say, sort of less traditional pathways about prefrontal cortical and ventral hippocampal inputs to the nucleus accumbens. And so they're not circuits that are traditionally thought of in the context of stress response, but they're pathways that we found are modified by stress. And then coming back to ask sort of what their function is and what they're doing, we've found that they do seem to be quite important in processing specific aspects of threatening stimuli.
What would be some examples of that? Like what aspects?
Yeah. So what we found, so in our most recent work, we kind of took a very reductionist approach, and we're just looking at which neural circuits process an auditory stimulus that predicts a foot shock. So the threatening stimulus is the foot shock. We can look at just the neural response to the foot shock, and we see that both prefrontal cortical neurons that project to the accumbens and ventral hippocampal neurons that project to the accumbens, both of these show an acute increase in neural activity to the foot shock. There are some really interesting sex differences there, but maybe let's come back to that in a second. But then when we can also look at how does that auditory cue that precedes the foot shock, how is that encoded? And we find that, again, both of these circuits encode that. But when we go a step further and we look at what's the response to a different auditory cue that the animals encounter in the same setting, but that's never associated with foot shock. So we can think of it as a non-threat cue. And we see that these pathways seem to encode the identity of that non-threat stimulus as well, but in these sort of curious sex-specific ways. And another sort of nuance to this work that sort of resists simple explanation is that we see that this neural encoding is happening when animals are just encountering these stimuli. But when we look at how that relates to behavioral responses to threat, and so remembering that there are lots of different ways that animals can respond to threat and can respond in a stressful or threatening situation, we see that neural encoding doesn't seem to have much to do with these sort of canonical threat behaviors like freezing, but it seems to be really important in how animals kind of use that information to guide sort of ongoing reward-seeking behavior.
Okay, so if I'm starting to understand this, basically something is going to end up being stressful, which means it's going to end up activating the HPA axis. But depending on exactly how directly stressful it is, or whether it's not stressful, but it predicts something stressful, like the tone paired with the foot shock, there are sort of multiple paths in the brain, multiple circuits that can eventually get you to the activation of the HPA axis. And there's probably different, I think what you started to say there is there's differences between animals that affect things like how easily new stimuli are going to end up affecting that stress circuitry. So for example, if you pair the tone with the foot shock's obviously a stressor, the tone is paired with it, so the tone sort of ties into things. But then it sounded like you were saying that if you just play like another tone, not that tone, but maybe a similar tone, for some animals, they say, okay, maybe that one's stressful too, but some animals don't do that. So there's this sort of susceptibility issue that seems like it's coming in here.
Yes, but I think there's kind of a couple of things to unpack here. And the first thing is sort of this comment about how different brain circuits sort of lead to activation of HPA axis. One thing to note is, like, in our recent work, we haven't actually been looking at this question of how HPA axis is activated. We're looking more at how, say, threatening stimuli are encoded. And then one of the consequences of that encoding, you would imagine, is activation of HPA axis, but we haven't looked at that directly. So just sort of put that aside for a second. But then I think the more interesting thing that came up from your comments is thinking about how, like, different brain circuits can encode different kinds of stresses. And I think this relates to something that is both interesting and very frustrating about stress as a stress researcher. And that is that it's very, very difficult to sort of settle on like a definition of what's stress, what's a stressor. And that's really the nature of the experience, that stress is highly subjective. And we can define a stressor, something that elicits a stress response, by this sort of outcome, how it changes behavior, how it changes cortisol release. But thinking about sort of how stressors, like the stimuli that elicit stress responses, are processed in the brain is going to be highly variable, because the things that elicit stress are highly variable. So you can have a stress response, you can experience threat from a tiger jumping out at you in the forest, but you could be sitting in your lounge room and just imagining something that's never happened.
Right.
And find that stressful.
Yeah.
And
Watching
so if
a
you
movie,
think about
whatever. Watching a movie, yeah.
Or even in the absence of any stimuli, just imagining, I have to do this exam next week. Right?
Yeah.
And so just appreciating sort of how variable the things are that can elicit a stress response. And I think it follows that the neural circuits that encode those kind of stimuli will be highly variable.
Right. So, I mean, we all can think of examples in our lives. You know, some people really lean into stress. Some people are stressed out by seemingly everything. So, in other words, the same stimulus can elicit a stress response in one person, but not in another, or the magnitude could be different, et cetera.
And then I think also how people react to that stress is enormously different. Like, I think some people really seem to thrive on stress and almost can't get motivated without it. And some people find any amount of stress to be disabling. So there's many levels of individual variability at sort of different parts of the process.
Yeah. And so all of that sort of, it speaks to this idea that the way that stimuli are encoded prior to touching the HPA axis is going to affect whether or not that axis lights up or lights up a little bit or a lot or whatever.
And then downstream, once the HPA axis is activated, that will have very different consequences for different people.
So thinking about like the activation side here and some of the circuits you were talking about, I'm going to ask a bit of an intentionally naive question. So you mentioned the nucleus accumbens as being an important part here. Normally, when we talk about the accumbens, a lot of people just associate that with reward learning and addiction. They don't necessarily associate it with stress. Was it surprising for you that this ended up being a place you were looking at, or why do you think that that's a circuit that's relevant here?
Yeah. So the way that I came to it was through work that I did in my postdoc. And so within the lab where I did my postdoc, there was a history of studying the accumbens in the context of addiction and animal models of and stress. And so there was already like a lot of papers showing changes to the accumbens with stress. So in that sense, it wasn't so surprising, but I agree with you that generally the accumbens is thought of as this reward center. But one way that we can think about sort of why that might be touched by stress is because when you have this experience of chronic stress, one of the sort of key functions that can be impaired is your ability to perceive and to use information about reward to guide behavior. And so I think that's one way we can understand the role of the accumbens in these stress-related disorders.
I mean, naturally, I start to think about, when I think about reward and stress, oftentimes the natural response we have to stress and chronic stress is to engage in reward-related behavior. Some people get stressed and they start eating junk food or whatever.
Yeah.
So there is sort of an intuitive tie-in, I would think.
And actually, this is not my work. This is a published work. I don't remember the group, but there's a really interesting paper showing that just giving mice sucrose is sufficient to dampen the activation of the CRF neurons in the PVN, the paraventricular nucleus of the hypothalamus, to dampen HPA axis activity. So probably not a great long-term strategy, but it does suggest that there's like a physiological drive there that consuming sucrose, and this paper only explored sucrose. You could speculate quite likely sort of other high caloric foods might have a similar
comfort food idea.
Yeah, that it is actually dampening HPA axis activation, at least acutely.
So basically, you give mice sugar, and the cells in the HPA axis that release some of these stress hormones are actually less active, basically.
Yeah. Yes.
Interesting. One of the areas I want to get into is individual variability and susceptibility. I know that there's a lot of work out there on some of this, and I'm only partially familiar, but I think a big tie-in here is going to be that a lot of early life experience affects things like the sensitivity of the HPA axis and how much it responds and how quickly it shuts off, whether or not the mother mouse was constantly licking the pup or not, whether there was many siblings competing for milk access, all of these things probably tie into this. But what can you say about the origins of variation in the susceptibility that individuals have to stress?
Yeah, I think this is a huge question. So I think we can all appreciate that individuals show differences in their susceptibility to stress, but why is that so? And I think there are lots of different factors, particularly in humans. We can't discount genetic factors. There's certainly genetic variation that plays a role in this. But definitely life experience is one of the factors. And so, I mean, there's epidemiological work in humans showing impacts of early life adversity. But in rodent models, so I alluded to this work in my PhD, just this sort of natural variation in the amount of maternal care that rat pups experience in the first week of life leads to quite marked differences in HPA axis function. And so differences in the peak and the return to baseline in core release, also ACTH, another stress hormone in the HPA axis. And that then goes on to produce differences in behavior, so differences in anxiety-like behavior. I found some really sort of unexpected differences in how animals learn, and in particular, how animals learn under stress, that suggested that animals that sort of had the lower levels of maternal care early in life showed this paradoxical increase in learning and hippocampal plasticity under conditions of stress, although at baseline they seem to be impaired.
So is the basic idea there that because they had more stress early in life, their brain is sort of tuned in to learning better under stressful conditions or something like that?
So that's kind of like the explanatory model. There's these ideas about preparedness. I think they fall apart if you push them too far. But the idea is that there's this sort of programming that might happen, that if you sort of are born into a high stress environment, you're going to thrive in a high stress environment. I don't think the epidemiological data holds up so much when you look at that. I think there's also a pretty good argument that like compounding stresses actually lead to worse outcomes. But yeah, that's one interpretation of this idea of why you have sort of stress enhancement of plasticity.
And, at least from the rodent work, it sounds like again, if we're talking about rodents, right, we're not talking about
Yeah.
anything
Yeah.
as dramatic as like the major human stressors we might talk about, like being born in a
Yeah.
war zone or into poverty or something.
Yeah.
Just natural sort of normal variation, like a mother mouse has a litter. There's some number of pups in it. I imagine,
Mhm.
if she's got a
Yeah.
particularly large litter, there's only so much time she can give to each individual mouse. And there's natural variation in how much licking each one gets, how much
Yeah.
cuddling and milk suckling
Yeah.
each one gets. Those types of things are relevant here. That sort of natural variation in early life experience leads to changes in stress
Yeah.
response.
Yeah. So actually, one of my colleagues in the Netherlands, when I was a grad student, did that experiment. It was absolutely heroic. So not only did she observe the rat sort of maternal behavior, I think we used to do it like 5 times a day for 7 days, something crazy. She did all of that, but she also like labeled all of her pups so that she could track exactly which pup was getting the care at each time.
Mm-hmm.
And sort of the upshot of that study was that, yeah, it seems that even so before she did that study, we just looked at the level of the litter, so that little rat family, if you will. But she took it one step further and looked at within litter variation and found that, yeah, it holds up there. So it does seem to be about the amount and quality of care that each individual pup experienced.
Mm-hmm. And naturally, I mean, one human thing this immediately makes me think of our birth order effects. Those of
Yeah.
us from larger families know that, even though you all get quote unquote the same treatment, there's only so much time in the day. And if you're the firstborn versus the
Yeah.
second or third or fourth born, you're just going to get a different level and quality of attention from your parents. And
Yeah.
naturally, that's going to affect your development.
So yeah, as a parent of multiple children, I have to say that I am a believer in benign neglect. So not all care is I think we have to sort of see the nuance in this, that it's not
Yeah.
just that we should give all of our offspring more care. Um, but I think the other thing to keep in mind, which I think is super interesting, is that you can also have sort of a converse where you have uh, children or individuals that have like objectively the same experience and yet have different outcomes. And I think
Yeah.
this speaks to the interplay between like gene and environment interactions,
Mm-hmm.
or perhaps some kind of like latent differences that emerge from like prenatal differences.
Yeah. Yeah.
Yeah.
And it sounded like there are some raw genetic, there's some raw genetic variability that might be relevant here. Is there much genetic variability in things like, I don't know, the genes encoding the cortisol receptor or whatever?
That's a great idea. I haven't looked a lot at this because we're working within inbred mice. And
Mm-hmm.
so we really kind of put genetics aside.
Yeah.
Um, yeah.
Yeah. So, I mean, independent of any genetic factors here, the fact that you see variability in inbred mice
Yeah.
means that there's lots of other things going on.
For sure. And so, like, where does that variability come from in our
Yeah.
mice? I think that it could be related to, as you say, differences in birth order, differences in utero positioning, for instance,
Yeah.
differences in the amount of care experienced early in life. So it could be essentially just the combination of all of the different experiences. But I think there's also an interesting possibility that there could just be stochastic changes
Mm-hmm.
in brain development.
Right.
That kind of start on a path that then becomes kind of canalized by later experience.
Yeah, that's an important concept. I've covered it before with people like Kevin Mitchell, but can you unpack that just a little bit more for people? What exactly do you mean by a stochastic developmental effect as opposed to, say, a true environmental difference between two individuals?
Right. So where we're talking about the effect of an environmental manipulation or some kind of environmental experience, we are thinking about the effect of a specific stimulus that somehow impacts the brain. So it has an effect on the brain, but there is an external stimulus. But with stochastic differences, essentially, largely in the absence of the input of specific stimulus, there's just some kind of differences. So we could just think about, say, the normal developmental processes unfolding, that maybe there's just some kind of variation, perhaps in the timing of that, in how different genes turn on and off, or perhaps in the growth rate of different sort of neuronal projections. So there could be all kinds of different biological processes just that are unfolding, and there's sort of some small variation in that could lead to individual differences.
Mhm.
It's not something that I've researched, but I think it's always important when we're trying to understand a mechanism that we kind of think widely about what it could be that's not what we think it is,
Mhm.
right? Because it's very
Yeah.
easy to convince yourself, oh, the evidence supports my idea. But I think we should always be thinking about, but what else could it be? Because we're generally less disciplined about looking for evidence for our unfavored ideas.
Yeah, and I think when it comes to thinking about things like developmental stochasticity,
Yeah.
our minds as just animals in the world, they're naturally tuned into looking for cause and effect and
Yeah.
agency and A goes to
Yeah.
B. What you're saying is, you've got genetic influences, and we all know what that is. You've got environmental
Yeah.
influences. Maybe mom was eating a different diet for me than she was for you, et cetera, et cetera.
Yeah.
But also, there's just random differences that
Yeah.
crop up based on a neuron going this way versus that way
Yeah.
for sort of purely random
Yeah.
mechanistic reasons.
Yeah. Exactly.
What about so another element to the variability and susceptibility to stress? So there's this sort of developmental stochasticity thing. There can be genetic variability between individuals. There can be these sort of birth order and other environmental effects that affect you versus a sibling. What about the role of sex here? I know that there's quite a lot of differences between males and females across species in terms of at the population level between the magnitude of the stress they experienced, how susceptible they are to certain things eliciting a stress response, et cetera, et cetera. What can you start to tell us about male female differences here?
Yeah, I think that there are profound and at least until recently underappreciated differences. Um, and so in our research, we talk about the effect of sex because we're studying mice and they have sex, they don't have gender. But when we're studying humans, we also need to think about gender. And that is this additional level of complexity, thinking about the social construction of gender identity. So I think the simplest thing to say is that we know that there are interactions between sex and acute stress response, but also in susceptibility to chronic stress. And so this is something that we've grappled with a lot in my lab in thinking about how to study both males and females. And so not even explicitly looking at sex differences per se, but just studying both sexes. And what is immediately apparent is that doing the same thing to males and females doesn't lead to the same outcome.
Mm-hmm.
And so this could be, say, if we apply like a stress protocol of, say, something like chronic variable stress, we know from work done by Georgia Hodis and Quincy LaPlante some time ago that females show susceptibility, so they show behavioral change after just 6 days of chronic variable stress. And this is sort of repeated experience of, say, foot shock, tail suspension, and restraint stress.
And when you say chronic variable stress, you mean so the stress is chronic, meaning it's going to last over a fairly lengthy period of time, but it's variable, so they don't really know when the stress is coming.
Yeah, so the chronic is that it's repeated, and the variable is exactly the stress that happens each day. It differs. So that particular paradigm is a pretty simple one where each day the animals are exposed to a stressor for 1 hour, and the identity of that stressor, that specific manipulation, varies each day and sort of just rotates every 3 days. So after 6 days of this, females already show behavioral change. So they show like increased anxiety-like behavior. They'll avoid the center of an open field. They show reductions in social interaction. But male mice take 28 days to show similar levels of behavioral change.
I see. So they get to the sort of same output, but it takes a much different period of time.
Yeah, exactly. And so then that in itself is interesting, and we can study the mechanisms for that. What we've been doing in my lab is asking questions about, so we had a paper looking at the state of susceptibility in males and females, where we were asking if we can induce kind of similar behavioral levels of susceptibility, do they have similar or different mechanisms? And so there we use different lengths of stress to induce similar levels of behavioral change in male and female mice. And then we looked in the brains to see, with this similar presentation of susceptibility, do we see the same neural pathways being recruited? And we do. Yeah. So this is a paper where we found that neural activity in the ventral hippocampus to the nucleus accumbens, so this same pathway that we keep coming back to, we found that exposure to chronic variable stress, so we actually used 4 days in females or 21 in males, increased activity in this pathway to similar levels in males and females, despite this sort of differing amount of stress. And looking into the mechanism of this. So first we found sort of this increase just using fiber photometry, which is this in vivo imaging technique, but using slice electrophysiology to try to understand the synaptic mechanism. We found that it seemed like what was happening with chronic variable stress, be it 4 days or 21 days, was increased in presynaptic input to these neurons in the ventral hippocampus that project to the accumbens. So this would sort of lead to the increased activity. But so what was really striking about this is that, I mean, there's a huge difference between 4 days and 21 days, right?
Yeah.
But we saw the same mechanisms being recruited.
Yeah. So it's as if, very vaguely speaking, it's as if the circuit is, keeping track of the total amount of stress. And if the stress gets big enough or quote unquote too much, it kicks in this way. But, how much is too much really depends on, in this case, if it's a male or a female.
Yeah, I think that's an interesting way to think about it. And it also ties into sort of another observation that we made, which is that with this technique in vivo fiber photometry, we were able to do rail recordings longitudinally. So we actually recorded activity in this neural circuit before animals ever encountered stress, and then across the course of stress and after stress. And we found that there were actually individual differences in the amount of neural activity in this circuit before stress that were predictive of that sort of behavioral presentation of stress susceptibility post-stress, which I think sort of comes back to this idea that you're suggesting that neural circuit might be kind of keeping track of stress. So you could, maybe it's some kind of readout of some kind of integrated susceptibility factor.
Yeah. Interesting. And that would vary between the sexes naturally, and also vary between individuals because of, all of the myriad things we talked about before.
So another interesting thing about that neural circuit, so AJ Roberson found around about the same time we published this paper, he actually looked at the excitability of these neurons in male and female mice, and found that in female mice, these ventral hippocampus to accumbens projecting neurons are actually more excitable. So they're more easily activated. And that's something that we've since replicated in our own lab. So we have individual variability within sex, but we also have this considerable variability between sexes.
And do we have any sense of where this arises? So, one thing that starts to come to mind here, the fact that we see this individual variability and we see it at the level of males versus females, one thing that comes to mind here is, I'm fairly familiar with some of the hypothalamic literature on the effects of estrogen, especially across the life cycle of females, like before and after pregnancy. And, as you probably know, as a mother, right, pregnancy often comes with changes in your sort of overall baseline levels of stress and anxiety. And it sort of makes ecological sense, right? Because now you have to be tuned into potential stressors in the environment because you have the little ones to look after. Are things like sex hormones involved in modulating the excitability of these circuits? How do we start to think about the origins of this difference?
So the short answer is yes. Yes, they are. So we recently actually got a grant to explore this in detail. So we haven't done much work on this as yet, but there's pretty good reason to think that it will be modulated. So actually, AJ Roberson, the colleague I mentioned, he did some interesting work where he actually pinned down this difference in excitability in the ventral hippocampus to accumbens neurons. He pinned it down to an effect of testosterone, not estrogen. And that it seems that in male mice, this androgen signaling is actually suppressing excitability. And if you do an orchidectomy, so removing the testes, this actually leads to increased excitability. That was an interesting effect, actually, because I think people often assume that all of the sex differences are driven by estrogen.
Right.
And certainly estrogen does a lot of things. But there's also effects of testosterone. And then I think another really interesting thing that is often kind of overlooked is that there's also estrogen signaling in males. Yeah, and it's crucial. Like if you don't have enough estrogen, normally we think of males as having quote unquote very little estrogen, but if you don't have enough, that can cause serious behavioral issues. Yeah, yeah. So in work that I did in collaboration with my postdoc lab, we actually found that estrogen signaling in the accumbens, the nucleus accumbens, seems to be protective against the effects of chronic stress in both male and female mice.
Hmm. Hmm. And I guess directionally, the effects you just mentioned about testosterone, it reduces the excitability of some of these cells.
Yeah.
That makes directional sense with how people normally think about testosterone. In general, the higher your testosterone levels, the more, I guess, impervious to stress the animal seems to be. Is that generally true?
Yeah, I don't know. I haven't really done any research on that to have an informed opinion. I think testosterone is going to have complex effects. And I think the other thing to keep in mind is that um, I think that while some of the effects might be sort of fairly similar in terms of changes in excitability or changes in spine growth or synaptic plasticity, uh, where in the brain testosterone or estrogen is acting is also
Mm-hmm.
going to have a big effect because of how neural circuits work, right?
Yeah. Yeah. And I guess one thing that could be relevant there is, correct me if I'm wrong, but these things are not just released systemically, they can also be synthesized and deployed locally in certain parts of the
Yeah.
brain.
Yes. Yeah. So you have local production of neurosteroids, which is fascinating, right?
Mm-hmm. Yeah.
Yeah. We need a lot more research into this, is what I would say.
Yeah. Yeah. Um, so what do we know about, okay, so we have individual variability and stress that people and mice and all sorts of creatures. There's variability in your susceptibility to stress, how much stress will manifest in terms of like your
Mhm.
behavior from a given stimulus. And there's difference between acute and chronic stress. So obviously, to some extent, some acute stress is normal and expected and potentially even good for you. But eventually, there's this threshold you cross where the stress becomes chronic
Mhm.
and it becomes unhealthy and maladaptive. And it's just too much.
Yeah.
One of the ways that can manifest is in chronic disorders like
Mhm.
PTSD, say.
Yeah.
What do we know about how individuals in a species transition from just responding to sort of regular normal amounts of stress to developing something like a disorder where now they're just having this hyperactive persistent over
Mhm.
response?
Yeah. I think one other thing to keep in mind is that the chronicity of stress is important,
Mm-hmm.
but also the intensity. And so, for
Mm.
instance, with PTSD could develop with sort of exposure to chronic stress, for instance, in personnel serving in war zones, right?
Yeah.
But PTSD can also develop from a single intense sort of near-death experience, right? And so it's about this complexity of the intensity of the stressor. The perception of control seems to be a really important thing, and then the chronicity. And so what do we know about how sort of individuals differently transition? So there's quite a lot of research now, largely in mice, looking at this concept of susceptibility and resilience. And so we can model this in mice, exposing mice to a chronic stressor. So one of the stresses we use commonly is a chronic social defeat stress. And so this is this paradigm where uh, mice are put into the cage of a bigger, meaner mouse for a brief aggressive encounter.
Mm-hmm.
This is really quite brief, and it's then the experimental mouse is sort of separated from that mouse, but still in the same cage. But there's a plexiglass divider with sort of holes drilled through it, so that there's this very potent psychological stress of being housed next to
Yep.
this mouse that just intensely bullied
Yeah.
you, right?
They can see it, they can smell it. It's probably a very stressful place to be.
Exactly. Yeah. But there's no physical harm. So this model isn't primarily about sort of the physical harm. Um, and so this experience is repeated every day for 10 days. And then at the end of this, mice are put into a social interaction test. And so they're put first into an empty arena and they can explore it. And then they're put back in when there's a mouse in this interaction enclosure. And that mouse looks the same as the mice that it had these aggressive encounters with, but it's actually a new mouse. So it's the same strain, but it's a mouse they've never encountered.
Mm-hmm.
And so what's really interesting about the social interaction test is that you might predict that all of the mice will just avoid interacting, right? Because they've had repeated experiences of getting defeated.
Yep.
But we find that actually only a subset of mice show this social avoidance, and that there's actually a good third of the population that continues to seek out interaction. And these are termed the resilient mice.
Yeah. So they're naturally resilient. They're
Naturally
willing
resilient
to go explore the stranger again, even though they
They're
have
like,
bad
maybe
experience.
this time it's going to be different.
Yeah. And I know from talking to other people that this idea of resiliency is very consequential. So there's probably a lot you're going to be able to talk to us about here. But one thing that I learned recently had nothing, this was from studies that had nothing to do with stress per se directly. But, I was talking to a gentleman from the Jackson lab, and he did these very, very large scale studies looking at calorie restriction in mice. And I don't need to get into the details here. I don't know if you've seen this, but the basic idea was, okay, you calorie restrict mice, you generally see lifespan extension, but you see a lot of variability between individuals within a strain and between strains. But one of the things they sort of happened upon was that, when you restrict mice with their food, when you pick them up just to weigh them and do things like that, those are all stressors to the mice, right? A giant human monster is coming and picking you up and doing these things. And what they found is that the level of weight loss that mice displayed was highly variable in response to just being picked up and weighed and going through all those experimental processes. And what they found is that a good index for stress resilience was how much weight they lost in response to these stressors. The ones that lost the least were the ones that were most resilient. And in those mice, they actually found the largest lifespan extension effects. And so one of the interpretations of this was that stress resilience may have had something to do with things like, in this case, lifespan. So my general question for you is, what sorts of things does stress resilience in mice predict? So when you sort of cohort them from when you take the highly resilient ones and the ones that are not resilient, do these mice display very different levels of lifespan, other health markers, other types of behaviors we wouldn't naturally maybe think of?
Yeah, that's a super interesting question. We have done more work asking kind of the inverse of what predicts the resilient phenotype. Because of the sort of opportunity that opens up in that if we understand what sort of predisposes animals to be resilient, maybe we can kind of shape the course of the trajectory towards resilience. But there's a lot of work, and we've done some of this, looking at the mechanisms of resilience. In terms of things about lifespan, I don't know that anyone has done aging studies. I think super interesting question. But certainly there are people who've done a lot of work about sort of neuroimmune interactions, that it seems like susceptibility is probably more of a state of inflammation.
Well, let's talk about the origin of the resilience. So basically you said you do this experiment where, the mouse gets beat up for a second and then they don't have to interact with that mouse again. So they're not going to get hurt, but they can see it, they can smell it. And you said about a third of the mice are what you would call resilient. They're willing to go and socialize again. And some fraction are probably, off in the corner avoiding those mice. What are the resilient ones? What's going on under the hood for them? What's making them resilient?
Yeah, so lots of things. We've done a lot of work looking at sort of patterns of gene expression. One thing that sort of I found early on is that it seems like across a variety of brain regions, in general, at this sort of, if we just look, say, one day after this social interaction test, so fairly soon after the chronic stress ended, in resilient mice, there's actually a lot more changes in gene expression. And one way that we sort of interpreted this is that there's this sort of organized transcriptional response. There's adaptation to the stress, whereas in susceptible mice, there's fewer gene expression changes, and this could be reflecting to sort of increased variability. And so we looked at the gene expression data through another lens, which was co-expression networks. And so this is the sort of idea that genes don't function in isolation. In fact, genes don't do anything except make proteins, but it's easier to sort of talk about things at the level of genes. But we have these sort of organized groups of genes that mediate biological functions. And so we can look at how the sort of correlational relationships between groups of genes change with an experience. And we found that in resilient animals, we were seeing sort of up or down regulation of the same groups of genes, but in susceptible mice, we're kind of seeing a breakdown of these correlational relationships, suggesting that something about how transcription is being turned on in the susceptible mice is kind of disordered. And potentially this is lack of adaptation.
Yeah. It sounds like what you're saying is that in the resilient mice, when you look at transcriptional changes, you look at changes in the brain in terms of how neurons are turning off or turning down genes after the stressor, the resilient mice seem to have a more coherent response.
Yes.
There are patterns of genes co-activating
Yeah.
and inactivating in a sort of structured way.
Mm. Yeah, exactly.
Is there, I know this is a bit of an irritating question, but what kind of genes were they? If you had to talk about, were these genes related to like building synapses, or was there any kind of pattern
There's
there?
a lot of it comes down to synaptic plasticity. Uh, I mean, it's not the only processes that are regulated, but certainly like these are the ones that we followed up on. And at least a little bit of this is because of my bias in how I think about the brain. So I think about the brain in terms of neurons and synapses. Whereas if you were talking to someone else, they might tell you all about the microglia, which
Yeah.
also do things related to neurons and synapses. But
Yeah.
I tend to be fairly neurocentric in my approach, but at least I admit my bias.
And if so you see this kind of transcriptional response difference between the resilient and the non-resilient mice.
Yes.
And then if I ask you where that comes from, I mean, do we know anything there? Like naturally, I would start to think about, okay, well, if you can have this coherent transcriptional change in the one group and not the other, maybe there's some kind of epigenetic thing going on where their histones are a little bit more labile or something.
Yeah. And so for sure, you can go a step back and you look at epigenetic modulation. And then I think then the question is, well, what caused the epigenetic changes? We can go a step back and a step back and a step back. And then we can sort of go back to like, well, is it early life? So there's sort of different levels of mechanism we can look at. Another one that we've looked at is just sort of looking simply at if there are sort of pre-existing differences in how neural circuits are recruited during experience that predict resilience and susceptibility. So I already told you that we found that there are differences in this ventral hippocampus to accumbens pathway that predicts susceptibility. Before that, we did a study looking in the nucleus accumbens, and we just looked at that. So there's sort of two main cell types in the accumbens, the dopamine D1 expressing and dopamine D2 expressing medium spiny neurons. And there are sort of changes to both of these after stress, but we were interested to see were there perhaps pre-existing differences in activity in these cell types that might predict how animals adapt to stress. And we found that if we sort of did this technique of recording before animals experience stress and then putting them through stress and identifying our susceptible and resilient mice, we found that the mice that would go on to become resilient seem to have increases in activity in the D1 medium spiny neurons, specifically during social interaction before they encountered stress. So there was some difference in how the neural circuits were sort of processing their social stimuli before animals encountered stress that was predictive of this outcome to stress. And in this study, we also looked at if maybe their behavior in that social interaction test was predictive of how they adapted to stress later. And at least sort of with the gross measures that we were using, we didn't see behavioral prediction in that specific study.
Okay, so in other words, basically, you've got mice that respond differently to stress. Some are more resilient than others. But even before they encounter any stress that you're gonna have them encounter, some of these neural circuits are already behaving differently.
Yes, exactly. Yeah, and so that can come back to the question of why is that? And I think we can. There's a lot of evidence that suggests that early life experience might be part of it. The recent work that we've done, we've sort of taken this sort of reductionist approach, thinking about, so we know that these circuits, the ventral hippocampus to accumbens, and also the prefrontal cortical input to the accumbens, we know that they're changed by stress. But why might that be? Is it because they're encoding information about aversive events? And as we talked about earlier, like we find that they are. And so we could think that then it's perhaps this process of responding to stressful events and learning about these experiences that could also be somehow modulating the activity in these circuits into the future.
And when we think about, a lot of this stuff, naturally comes back to like early life experiences. When you're a baby or a mouse pup in the nest or whatever. Is there anything? So when we think about the development of these stress responsive circuits, the development of these circuits that are encoding, the different aspects of the environment that are related to stress, is there something like a critical period here where after some point the circuit is sort of set and the behavior of the mouse will be relatively consistent for the rest of its life?
So I think we can think perhaps less about critical periods and more about sensitive periods, that it's not sort of a the distinction being that a critical period is really like this absolute restriction on the opportunity for plasticity. So we see critical periods, say, in visual system development, but a sensitive period is that there's sort of more flexibility, more opportunity to reshape the system at certain times of life. And I suspect we're dealing more with sensitive periods rather than critical periods.
So instead of a defined window, it's more of just a gradient that slowly changes as the animal develops.
Yeah, it's more about sort of how hard that cutoff is in terms of the opportunity for plasticity. After, like, if it's a critical period, essentially there's very little you can do to change that plasticity. But if it's a sensitive period, it's easier to shift it in that window, but it's still possible. But I mean, ultimately, I think critical periods and sensitive periods probably exist more on a continuum than is suggested by the way that we talk about them.
Okay.
What I would say is that I think that there are, say, windows of heightened sensitivity that most likely relate to sort of the time course of development of different neural circuits.
And when we talk about, it's easy to talk about too much stress. At some point, it's always too much for anyone. No matter how resilient you are, there can be too much at some point. What about too little stress? Like, in what sense can we say that stress is necessary for normal learning? If an animal experiences, no significant stress, does that ever cause problems? Is there a mouse model of coddling?
Intuitively, yes, but mice are pretty jumpy. So
okay. Yeah, they're little prey animals. So
they might not be the best model for studying that. But yes, I think like maybe the sort of the easy way to answer that is that, like, absolutely we know that stress hormones, cortisol, corticosterone, and also norepinephrine, have an adaptive role in sort of reinforcing synaptic plasticity. So, and you need cort to survive, right? So, for instance, in experiments where HPA axis function is being investigated and you remove the adrenal glands to prevent that sort of HPA axis activation, it's still necessary to replace with a constant amount of cort.
So, what do you mean necessary? What happens if you don't do that?
I guess ultimately the animal wouldn't survive. But
is it just because it's not responding to threats and getting away and getting to safety, or what?
No. Even a lab rat that can sort of live a pretty cushy life needs cort because of the diverse functions that cort mediates.
I see. So this is probably another area where our language can mislead us. We think of cortisol as a stress hormone, but it's doing other things other than a pure stress response.
Yes. Yeah, it has really diverse functions.
What are some of the other major things that it does?
It has a lot of metabolic functions and just general, like, energy balance. Yeah.
Interesting. And I don't know if this is your area, but I know this is something that comes up quite a bit, that, you mentioned earlier that there are these metabolic effects that are inherent to this stuff, because a stress response needs to change how an animal's mobilizing its energy stores in order to help it do the behaviors it needs to deal with the stress. But, we know that there's a lot of chronic stress in human societies today. And I know that some people think that that's very much tied to things like metabolic syndrome. How much do you know about the role of chronic stress in things like metabolic disorders?
Yeah, so it's certainly not an area, it's not my area of expertise, but as an interested person, certainly I'm aware of this link, that chronic stress does also lead to metabolic disorders. And there's also an interesting intersection of metabolic disturbances with mood and anxiety, right? And so there's this sort of, could think of it really as this, like, downward spiral of metabolic changes, perhaps induced by poor diet, leading to this sort of aggravation of mood and anxiety, which is quite likely going to reinforce those kind of dietary choices.
And so when we think about sort of the link between some of these mental health issues, so we can talk about stress and the classic stress response, and then you've got things like PTSD, you've got things like major depression or anxiety disorders. These things are all quasi similar in certain ways, but they're all different. What are some of the differences between how why would in some individuals, what do we know about how chronic stress exposure leads to, say, a depressive phenotype in some set of individuals, but it leads to more of a PTSD or like phenotype in others?
Yeah, that's a really interesting question. And I think one way to think about this is really coming back to the question of, well, what is depression? What is PTSD? And are they things? Are they absolute things?
Right.
And I will leave that to the psychiatrist to decide. But I think that there's increasingly an appreciation that, from a research perspective and probably from a treatment perspective, it's more useful to focus on specific symptoms. And then you could sort of decompose specific disorders into their symptoms. But more and more, we've been kind of just taking the approach of, instead of thinking about a model for depression, we're studying some specific function that is relevant for depression, but is likely relevant across a lot of disorders. So we've been doing a lot of work, this is unpublished work, looking at reward processing. And so this is something that's really important when we think about sort of anhedonia, this inability to experience pleasure and to use information about reward and depression. But there's also changes in reward processing in other disorders like PTSD, schizophrenia. So why do people sort of develop the specific symptom profiles that they do? I think this is going to be a very complex question to answer that is almost certainly going to be the intersection of their experience, their genetic makeup, and the situational factors. Yeah.
Another element of stress susceptibility I wanted to ask you about is obviously there can be genetic differences between people. You get around that largely with the mouse models because these are inbred strains that don't have a lot of genetic variability. Nonetheless, there's a lot of individual variability, and we've talked, at length about early life experience and these things, and all of that's pretty intuitive. Obviously, a variation there is going to translate to individual variation in stress response. Are there any? When we talk about the stress response generally and susceptibility, are there any transgenerational effects such that if, say, your mother or father experience a lot of chronic stress, say, can they pass that on through epigenetic means?
Yeah, this is a very hot topic and enormously controversial. I think the first thing to appreciate is that people mean different things when they talk about transgenerational epigenetic inheritance. And something really important to appreciate is that a lot of studies that look at mechanisms like this don't actually go enough generations to be looking at this true epigenetic inheritance. And so this is because a female fetus already has all of its eggs in utero. And so when the mother is exposed to some kind of experience, the fetus is being exposed, but the eggs in that fetus are also being exposed.
Yeah. The next generation is already there in that sense.
Yes, exactly, which is kind of wild, right?
Yeah.
And could be an enormous weight to all mothers, but also good to know that there's also evidence of paternal transmission through sperm. So fathers aren't completely off the hook. So I think it's a really interesting area of research. I think definitely more work needs to be done. And I think that the field's really interested to sort of learn from this work and really assess the evidence.
Yeah, and this is an area that gets a lot of attention, just generally speaking, this idea of epigenetic inheritance and stuff like that. I think what you're starting to say, which is important, is it's not enough to simply it's not enough to do some manipulation to one generation, to say expose an animal to a stress or a different diet or whatever, and then measure that their offspring were affected by that and say it's a transgenerational effect, because what it could be, which is subtly but distinctly different, is it could have been an exposure of that generation itself to the same manipulation, because those little eggs were already there.
Exactly. Yeah. And then I think the other thing to keep in mind is that there are sort of also behavioral mechanisms of transmission, right?
Right.
That the behavior of the parents can lead to sort of programming of the offspring that then leads to this behavioral transmission.
Mm-hmm.
So,
which could be easily misinterpreted as a transgenerational epigenetic effect.
Exactly. And so within animal studies, there are controls to do, like cross-fostering controls going to the F3 generation. But this work is really demanding and time-consuming.
Right.
But we need more of this kind of work to really establish very strong evidence of this.
Yeah. What are some of the ongoing experiments in your lab right now? What are the questions that you guys are asking at this moment?
Yeah, we've got a lot of different directions. So I mentioned that we're doing a lot of work about reward processing, looking at, we have a sort of a paper in revision right now that's looking at our same favorite ventral hippocampus and prefrontal cortical inputs to the accumbens and how they're processing reward outcomes. And really interesting finding is that not only that they're processing threat, but also reward, but that there's absolutely no sex difference. And so this kind of comes back to supporting our interpretation of our earlier work, that where we saw sex differences in the neural circuits that were encoding threat, we ultimately concluded that that was because there was very subtle differences in behavioral strategy that were leading to recruitment of the circuits differently between males and females. And so finding that within this reward context, that we don't see any sex difference in the neural circuit recruitment, supports this idea that it's something specific to how males and females are processing threat that leads to different recruitment of these circuits.
So when you see something like a sex difference that appears to be a difference in reward processing or stress response, it could actually reflect a difference in the behavioral strategies that lead to those responses, rather than like an intrinsic difference in the reward processing per se.
Exactly, yeah. So we saw the difference in threat processing, and ultimately what I think is going on is that in the female mice, there's this sort of subtle difference in how much they learn about the context as a predictor of threat. And so that leads to this kind of overall higher level of threat perception that then enables them to learn that non-threat cue that we talked about, the auditory cue that predicts that no shock's going to happen. They learn that more as a safety signal. They learn, okay, when that cue's played, I'm not going to get shocked. Whereas the male mice seem to just be learning about the threat stimulus. This cue means I'm going to get shocked, and they largely ignore the other stimulus. And so that's a subtle but important difference, where the non-threat cue, by sort of on average, males are ignoring it, but females, because they have this higher level of contextual sort of threat learning, learn this safety association. I think this is probably why we have different recruitment of the circuits between males and females. But the interesting thing is that it really is situationally specific, and it's suggesting that experience interacts with neural encoding. And this is something that we sort of see again and again, that we look at how the brain encodes experience, but the experience is also sort of feeding back to influence how those neural circuits are being recruited.
And you mentioned before that you're a mother. So given that this is your field, and you talk and you study all this stuff, and you mentioned something earlier. You mentioned benign neglect, and that can have its place. What has like doing all of this as a scientist, how has it shaped like your ability to think about stress in early childhood and your interactions with your children?
Yeah, it's an interesting question. I think I like to think about the opportunities for resilience. And I think that this is an important point, because it could be easy to walk away from this kind of research and think that we're all doomed. But I think resilience is a really powerful concept. And I think a really important thing to appreciate about resilience is that resilience is a phenomena that emerges through exposure to stress. So in the absence of stress, there's no resilience. And so in some ways, we can think about stress as opportunities to learn about, to develop resilience, right? But for that to happen, the stress needs to be sort of controllable and not overwhelming. So I think
limits on intensity, limits on how often you're encountering it, all of these things.
Yeah, I think the big thing is about sort of perception of control and the ability to sort of master that challenge. And so this is certainly something I think of as a parent, is that my goal is not to give my children a life that has no stress. It's about helping to give them the tools to deal with those challenges and to meet those challenges to develop resilience.
Mm-hmm. Do you have any like final thoughts you want to leave people with, or anything you want to reiterate about all of the ground that we covered today?
Yeah, I think we covered quite a lot of ground. I think one of the things we talked a lot about is about individual differences. And we talked about this sort of a little bit in the context of sex differences. I think it's always helpful to remember that there are sort of many different axes of individual differences, and that as researchers, we should be thinking about this. We should be thinking about the phenomenon of individual differences, and that the mean, sort of the group average, is not representative of most individuals, if any, and that we need to be open to this variability and understanding that to really understand biology.
Mm-hmm. Yeah, I think what you're saying is, obviously there's sex differences, there's males and females, but there's also birth order, there's also all of these other factors. And sometimes, sometimes it's just easy to overlook those. I mean, we don't normally think about being the third born mouse in the litter as an axis of variability.
Yeah. Yeah. So just keeping in mind that biology is complex and being open to that, to the variability and the nuance.